I don't know what to do, Doc. My legs are hurting, I have the chills, and I'm failing biotech.
Son, I don't know how I'm going to break this to you, but I think you have a genetic disease.
What should I do?
Well, son, we have a new secret weapon.
Give it to me straight, Doc.
Look, here he comes now.
He?
I'm the secret weapon.
Snoop Dogg. You know what happened with the D.R.E.
I'm the gene man. So what is gene therapy, you ask? So gene therapy is the process of treating a disease or disorder by replacing a dysfunctional gene with a new functioning one. So this is done using the use of recombinant viral technology. It's on like the forefront of scientific discoveries. And it has the ability to help diseased tissues and organs work properly just as new. This technique it's risky, and it's still being tested to ensure its safety and efficacy. And it's really being focused on diseases that don't have a cure that's being marketed right now. And how this is fundamentally different from traditional drug-based treatments is that it fights the underlying genetic issues, not just the symptoms. So this is an example of gene therapy in action. So this is a faulty gene. This is a Calvin Klein gene. We don't need that in our DNA, so we're gonna take that out. We're gonna cut it out using recombinant procedures, and we're gonna put in new DNA, and it's as simple as that.
That all sounds good and peachy, but how'd you get here?
Oh, there's my ride.
Oh, you got here through me, bro.
Low life, yo life, boy, we living it up. Taking chances while we dancing in the party for show. Slip my
You may ask, nobody got nothing on me. You're asking how to introduce the delivery systems with gene therapy? I got you. You can use transduction, which uses viral vectors. Yeah. Let me just spice it up a little. There are two types of viral vectors that you use while using transduction. It's integrating and non-integrating. With integrating, you use retroviruses, lentiviruses, and adeno-associated viral vectors. With retroviruses, you use RNA viruses to carry out the genes for reverse transcriptase. Oh yeah, dude, it's cooking. With lentiviruses, you replace those genes for glycoproteins from another viral genome in the packaging cell of the vector. And then the last one, there's a number 3, use adeno-associated viruses, which target non-proliferating cells and have discrete genome inserting sites. Check this out, my Jordans. And then for non-integrating, it's pretty much like using, it's maintained in the nucleus without integrating into the chromosomal DNA. So it's like pretty much not integrating, just a word. Yeah. And then the other way is transfection, which is pretty much like non-viral vectors. And then to use that, you can use plasmids, you can use the gene gun, and then you can use liposomes. And that's the way I got here.
Whoa, why does this sound so far out?
Well, calm down there, buddy. First, let me tell you what gene therapy can be useful for.
Compton, Long Beach, Inglewood, South Central out to the west side.
You. So, what is gene therapy useful for? Gene therapy has the potential to treat and cure diseases that are inherited or are genetic in origin. Research and development is in progress for several gene therapies to perfect and improve treatment for common conditions and diseases. Gene medicine trials have been already conducted on cancer diseases, monogenetic diseases, infectious diseases, and cardiovascular diseases. Now I know this is a lot of information, but I've narrowed down your disease to 2 possible possibilities.
We gon' rock it till the wheels fall off.
Hemophilia and Parkinson's. Hemophilia patients are missing proteins that help clotting factors. They suffer from internal and external bleeding that can be life-threatening. Researchers were able to successfully use an adeno-associated viral vector to deliver a gene for the missing clotting protein in liver cells. So after treatment, most patients were fine, luckily. For Parkinson's, these patients lose cells that are associated with dopamine. Patients lose the ability to control their movements as disease develops. Small groups of patients were treated with a retroviral vector to introduce 3 genes into the brain. The genes gave some cells the ability to produce dopamine, which it couldn't do before. All of these patients had improved muscle control after treatment. The first approach in doing this is to increase dopamine production in specific regions of the brain. One study using this approach uses the gene for the enzyme aromatic amino acid decarboxylase. The second therapy strategy is to adjust or modulate the excitatory and inhibitory pathways of the brain. The third approach of this is called growth factors because of their role in brain development, and they might protect against progression of the Parkinson's disease or possibly even reverse it by stimulating regrowth of injured nerve cells.
All right, now that you've heard all of our information, we gotta tell you, all I can say is wish you the best of luck, and we hope that gene therapy could be used to help a disease you're suffering from.
So here's your gene. Good luck, mate.
Always a mate. Ain't no party like a West Coast party, cause the West Coast party don't stop. So when you see a young nigga in a Chevy hitting switches, then you gotta get a nigga that's pride. I got size in my rise and the motion for your ocean. Coolio got the potion.